I think it is clear to believe in the power of ideas.
Fresh thinking at the Manhattan Institute.
I'd like to thank everyone for taking time out of their very, very busy schedules, I'm sure, to be here this morning. We have a terrific program. Let me introduce myself. My name is Paul Howard. I'm a senior fellow and director of the Manhattan Institute's Center for Medical Progress. I head up our healthcare policy shop there. And the Manhattan Institute, for those of you who are not familiar with us, we're a nonpartisan think tank, a 501c3, that advocates for market-based public policies and evidence-based public policies across a number of different issue areas, ranging from crime and education to, of course, healthcare. The Manhattan Institute's and Project FDA's interest in the FDA is that we see it as the locus of and gatekeeper for transformational medical innovation in the U.S., an agency that can help keep the U.S. biopharmaceutical industry as the world's most innovative and leading delivery system for breakthrough therapies that have the potential to not only save lives and improve lives, but of course lower healthcare costs, a topic that is on everyone's mind. Our FDA initiative, Project FDA, is led by former FDA Chairman, former FDA Commissioner, Dr. Andrew von Eschenbach. And I'll just introduce him briefly and then let him come up and say a few words about what we're trying to achieve here. From September 2005 to January 2009, Dr. von Eschenbach served as Commissioner of the U.S. Food and Drug Administration, where he championed an agenda to modernize the FDA. He also joined the FDA after serving for 4 years as Director of the National Cancer Institute at the National Institutes of Health. At the time of his appointment by President Bush to serve as a chairman of the National Cancer Institute, he was also president-elect of the American Cancer Society. Dr. von Eschenbach has held numerous leadership positions throughout his 3 decades of career as a physician, surgeon, and oncologist, including Executive Vice President and Chief Academic at the University of Texas MD Anderson Cancer Center, and he's published more than 300 scientific articles and studies. He received numerous professional awards and accolades, including in 2006, when he was named by Time magazine as one of the 100 most influential people in healthcare. Let me also say a couple of things about Andy, Dr. von Eschenbach, that you won't find on his official bio. It's been a real pleasure working with him, close to 2 years now. When you walk into a room full of people who are either patients, maybe they're senators, maybe they're industry, maybe they're FDA staffers, you just watch heads start to nod when Andy talks. And that is a real credit to the depth and breadth of his understanding of the system, as someone who's participated at every level as a caregiver, as a physician, as someone who's held leadership positions at very important hospital systems, and then again at the National Cancer Institute, and then at FDA. So here is someone who has seen the whole innovation ecosystem, understands it deeply, and can talk about it in a way that resonates with people who are living and fighting these battles every day. And I'd also like to say, from having been in these meetings with him and talking to people who've served with him at the National Cancer Institute or at FDA, and I think Andy would appreciate this metaphor, it's like talking to players who've played for John Wooden or Mike Krzyzewski at Duke. They're deeply loyal to him. They're passionate about what he was trying to achieve at those agencies, and speak in glowing terms about him. So it's a real testament to the man that when he serves in a leadership role, he generates that kind of engagement and passion from the people he's leading, which is tremendous. And one final interesting note about Dr. von Eschenbach, in addition to being able to talk about the finer points of clinical trials and drug development, he can also tell you the best place to get a cheesesteak in South Philadelphia. So please join me in welcoming our FDA Chairman, Dr. Andrew von Eschenbach.
Thank you, Paul. Thank you, first of all, for that gracious introduction. But you had promised me it was going to be short. You also had promised me you were not going to out me as a former commissioner of the Food and Drug Administration to also have been an expert on cheesesteaks. First of all, I want to begin by sincerely thanking each and every one of you for being here today. We knew in issuing invitations to this meeting that you come from extremely busy backgrounds and have enormous responsibilities. So for you to take the day to spend it with us, to engage in a conversation is a great privilege and a great honor for the Manhattan Institute, and we thank you for that. We also believe, of course, that it's worthwhile you having done so. It's important that we are here today. The Manhattan Institute has had a long-standing commitment to the interest of the Food and Drug Administration long before I joined the Institute and was privileged enough to be asked to head up the FDA project. And the reason for that interest and that commitment is because they, as well as you and I, recognize the core central importance of this one regulatory agency to almost everything and anything we hope for this country to accomplish in the first part of the 21st century. The 21st century is emerging as the century of the life sciences, just like the 20th century began with our exploration of the atom and its nucleus, and it created a trajectory for the 20th century. This century, we've been exploring the cell and its nucleus, and that knowledge is transforming everything: healthcare, agriculture, animal husbandry, energy, biodefense. And the one agency of all of the agencies within the federal government that sits at the nexus of all of those paths of opportunity is the Food and Drug Administration. So whether you're talking about human health or quality of life, or whether you're talking about economic development for this nation and its impact on the world, that conversation at some point in time must include a conversation about the Food and Drug Administration. And so the Project FDA is intended to create exactly the forum for that broader conversation. And I know from having, been in the seat and now recognizing the life of my successor, Peggy Hamburg, and what life is like for her in that seat, as soon as a group of people come together to talk about FDA, the immediate expectation is it's an FDA bashing session. The FDA project at the Manhattan Institute is not about bashing the FDA. It's not about continuing to criticize the agency regarding a particular failure of performance. It's to create a forum for a constructive, but yes, critical discussion of what needs to be done in order to continue to keep the agency in pace with the incredible rapid changes that are occurring in the world around it. Science and technology are driving society in an almost exponentially accelerating pace of progress. And that is changing and altering life around us. It's changing and altering our understanding of life. It's changing and altering the kind of products that we are developing, be they medicines or drugs or foods or whatever, that are influencing life. And so there needs to be a conversation about that critically important agency that regulates all of that, so that we understand it's a strategic agenda for the future. We're able to then intelligently begin to advocate for that strategy and for that agenda, be able to effectively support the agency in accomplishing that agenda, and basically begin to fulfill the promise of promoting and protecting the public health and the quality of life of the people we serve. So the FDA project is privileged to have you contribute to that process. I'm going to make this a little bit of a recruiting speech and ask you if you will, even after today, continue to contribute of your insight, your wisdom, your time, and your energy for this broader conversation that we have to have as citizens and as a nation so that we stay at the forefront of progress and prosperity in the 21st century. That's why you're here. That's why we've asked you to be a part of this, and I am deeply grateful for your ability and your commitment to contribute. So thank you. Thank you, Paul.
You can tell by the topic of the conference, the digital future of molecular medicine, rethinking FDA regulation, that we're not just talking about the agency, we're talking about the cluster of technologies that are growing up around drug development, outside even of what we traditionally think of as drug development in terms of players like IBM or other leaders that are developing these new technologies that have tremendous implications. One thinker at the Manhattan Institute who has been a real resource for us and a polymath in terms of understanding how new technologies are affecting the ecosystem not only of healthcare but of other fields is Peter Ueber, who will be speaking shortly. Allow me just to introduce him. Peter is a senior fellow at the Manhattan Institute, writing on the issues of drug development, energy, technology, and the law. He's the author of the forthcoming book, The Cure in the Code: How 20th Century Law is Undermining 21st Century Medicine. He most recently wrote The Bottomless Well, co-authored with Mark Mills, which Bill Gates called, quote, which Bill Gates said is the only book I've ever seen that really explains energy, its history, and what it will be like going forward. His previous book, Hard Green, Saving the Environment from the Environmentalists, was called, quote, The Richest Contribution Ever Made to the Greening of the Political Mind by William F. Buckley. Peter has written widely also about legal issues related to the FCC and junk science in the courts, which have been cited, in fact, by the Supreme Court. He's published dozens of articles in scholarly journals such as the Harvard Law Review and the Yale Law Journal, as well as popular journals like the Wall Street Journal, Reason, Regulation, and National Review. Before joining the Manhattan Institute, he served as an assistant and later associate professor at MIT for 6 years. He clerked on the D.C. Circuit Court of Appeals for Judge Ruth Bader Ginsburg, and then later on the U.S. Supreme Court for Sandra Day O'Connor. He is a partner at the law firm of Kellogg, Ueber, and Hansen, Todd, Evans, and Feigl in Washington, D.C., and he holds a doctorate in mechanical engineering from MIT and a law degree from Harvard University. I think Peter stands at the unique position of someone who not only understands the technology, he understands the cultural currents that are driving those technologies forward in the marketplace, and he has a unique gift for telling us in visionary prose how those technologies are going to be affecting our lives in the months and years to come. Please join me in welcoming Peter Ueber.
70 years ago, Alan Turing grasped that logical code is very good at decoding other codes, and he then went on to build the machines that cracked the Nazis' Enigma code. Today, our computers are quite powerful enough to decode major chunks of human biochemistry and understand how it choreographs our health and our diseases and our responses to drugs. And now our computers are being fed by torrents of new molecular information because there's a second revolution underway, the biotech revolution, that can read all of this stuff down there at the bottom. I think it's almost certainly the case that never before in history have two such powerful technological revolutions, digital and biochemical, converged to advance such a single such important objective. I intend to talk briefly about its impact at every stage of drug development, but without doubt, the FDA is the weak link in this chain. It is lagging far behind the other edges of drug development, the pre-FDA and post-FDA, in its mastery of molecular medicine. The thing to emphasize first here is that the FDA, I am a lawyer, the FDA doesn't actually license drugs. It licenses drug-patient combinations. The exact statutory language is that the implicit guarantees given in a drug license apply under the conditions of use prescribed, recommended, or suggested in the labeling thereof. So you are licensing the pair, not just the drug. And the FDA controls the key stage of the actual process of developing drug-patient science. You don't start working out that science until you put a drug, the real parts, the final parts, until you put a drug inside live patients, and that is meticulously controlled by the FDA. In fact, it requires a different kind of license, an investigational license. So this gives the FDA enormous influence on how fast we develop the power of molecular medicine and how well we develop it. And cutting to the chase, and then I intend to spend the rest of my time trying to persuade you that this is true, I think that it can be said that under the FDA's standard, rigid, scripted drug testing protocols, there are many drugs that we can develop today, we clearly have the power to do it, that we desperately need and want on the shelf that simply will never get through Washington because those protocols simply don't provide the flexibility and don't use the methodologies that will let that happen. So let's start at the beginning. Not quite 2 years ago, the National Research Council, at the behest of the National Institutes of Health, issued a very important report. It's called Toward Precision Medicine. And its objective is to discuss a new taxonomy of human diseases based on molecular biology. And in effect, it recommends that it says we have to redefine diseases completely, based not on clinical symptoms, as they have been since time immemorial, but based on the molecular structure of the diseases and the dynamics. And to that end, they recommend putting together what they call a knowledge network. I see Google fingerprints all over it. They actually cite Google and so on. They're saying basically we have to continuously pour in molecular and clinical data and build these very large databases that will begin exposing the links between the molecular foundations of diseases and the clinical effects, and throwing in drugs as well into that process so we gain new insights into how the drugs change this. Narrower versions of this are already, of course, underway through the NIH's Cancer Genome Project and the Human Microbiome Project and the 1,000 Genomes Project. It's already clear from what's already proceeding very fast that there is far more biochemical diversity in human bodies, far more than was previously thought, lurking under most of our diseases and thus affecting the performance of our drugs. For example, targeting epidermal growth factor in some lung cancer patients is a very good strategy, but we happen to know that there are 20 genetic variations in the EGF gene that codes for the proteins that we're trying to target. There are, with many diseases, multiple biochemical pathways to the same clinical end state. So if you start thinking clinical end states and begin looking for drugs, you're almost certainly going to be in for bad news because there are many different ways of getting to those clinical endpoints. Those multiple pathways sometimes operate simultaneously. I mean, in cancers often provide the most vivid illustrations of these things. Sometimes cancer, the cell proliferation is being triggered, but not by one signaling pathway, but by several. So if you shut down just one of them, you get some good effect, but not enough. There's more you have to do. We used to assume, the standard clinical assumption since time immemorial almost has been that a common disease reflected a common underlying cause. The new model, really rapidly emerging and gaining dominance is that common diseases often reflect many rare variations in the underlying chemistry, and particularly in genetic chemistry. And of course, once you begin dealing with drugs, all of this is further complicated by the problem of side effects, which can involve biochemical variations anywhere in the patient's body, and which just add additional layers of molecular complexity to your drug's performance. To pick one example, which is quite often significant, liver genes can affect how well the patient's body metabolizes a drug. Sometimes the metabolites are what are doing the real work, not the original drug, and then alternatively, sometimes if you're not metabolizing it well, the drug is going to poison you and you're going to get dreadful side effects. And we have already found quite a few biomarkers, molecular signatures that will tell you in advance, does the patient have the right liver to make this drug look good? There's nothing wrong with the drug. There's nothing really wrong with the patient. They just got the wrong liver for this particular drug-cancer combination. Now, start looking at this from the drug's perspective before we get straight to the FDA. Our biochemists, I may overstate this a bit, but not by much, are really incredibly good now at designing a molecule to whack another molecule. You give them a target, and at least in the lab, you say, go do something to it, and they'll almost certainly come up with one. They've got two very powerful tools for doing this. One is structure-based design, which won 2 Americans, and I think it was a Brit Nobel Prize in 1988. This was a year after the first HIV drug drug was licensed, and the two, Gertrude Elion and George Hitchings, had played a distant role in getting that drug into the line. And then, of course, the monoclonal antibodies are used for similar purposes. They're precisely targeted. You show the bioengineered mouse the target, and they'll come up with an antibody that'll target. During the HIV years, particularly in the 90s, according to the National Academy of Sciences, and I think they're quite right, had a revolutionary effect on modern drug design, this ability, this methodical way to look at a target and design an anti-molecule to hit it. I mean, in 2007, to pick one recent example, a lung cancer, the identification of a lung cancer gene in 2007 led to clinical trials 3 years later, and the FDA's accelerated approval licensing in 2011. 4 years from finding the gene to getting a drug to market. That's pretty good by Washington standards. Targeted drugs are a very good start, but often won't be enough. HIV and cancers require cocktail cures. They mutate so fast that one drug often can't do it. There are many strains of HIV have emerged and continue to emerge. They reflect sexual and lifestyle factors, and the virus is so nimble, it's really not a disease. It's an engine for generating an endless variety of diseases. Until recently, doctors were selecting, we have about 20 drugs on the shelf now to deal with HIV, and until quite recently, doctors would pick 3 of them off the shelf, kind of more or less by trial and error, and keep trying until they could find how to lower the loads in the individual patient. If you want to do it really well now, you don't ask a doctor, you ask an IBM computer. They actually build up a network in Europe, the Uresist database. When last I checked, it had the data from 50,000 patients, 130,000 treatment regimens, 1 million records of viral genetic sequences and viral loads and white blood counts. It's continuously updated, and by golly, those computers are really good at looking at patterns, and you give it a patient signature, and they crank out the right prescription. And just to prove it, they actually ran an engine versus experts trial. They took 20 case histories that the computer had never seen before, and the experts hadn't either, and the computer, this is Jeopardy all over again, the computer beat the experts 9-1. I think there's not the slightest doubt that give computers enough molecular and clinical data, and they will bridge that huge gulf, that huge divide better than any human being. They're just very good at patterns, and we've got tremendous people who are hungry for money and fame and glory and would like to save humanity now that they've made 50 billion or something too. They're happy, they're quite eager to do it, and they can do this better than anybody else if you feed them the data. Cancer cells behave much like HIV. They mutate fast, they adapt quickly to our drugs. They require even more complex drug regimens. IBM is inching into this as well. They've announced a new engine, and I think it will have great promise. Myriad Genetics is doing this. It's made, hundreds of millions on 2 genes for breast cancer. They're currently litigating one of those genes before the Supreme Court. 23andMe, there are a bunch of other companies all getting into this basic problem of how do we link the molecular to the clinical. There's a big gulf between them, and all of them basically set about in the same way. We generate torrents of data involving both, and we use digital technology to make sense of it all. In the race to precision molecular medicine, again, the FDA, as I said, I think is the very weak link. It's been thinking molecular since about the 1970s, when we began linking cholesterol and such things to a few very common diseases. It licensed the first targeted drug in 1981. It's been mulling over how to shoehorn molecules into its venerable trial protocols at least since 1992, when it passed its accelerated approval rule for HIV drugs. Congress codified and expanded the accelerated approval rule in 1997. But to this day, it applies it in a very limited and ad hoc way, almost only to HIV, exclusively to HIV and cancer drugs. And there's some talk in the agency, some reactionary talk of pushing back or even abolishing it. Last September, President Obama White House, which I confess I do not often cite for propositions that I like, that issued a wonderful report, a really good one. The Council of Advisors on Science and Technology, the Pcast Report, recommends the opposite. It says accelerated approval allowed for the development of pioneering and life-saving HIV, AIDS, and cancer drugs over the past 2 decades. It recommends that we systematize it and make full use of it for all drugs that address an unmet medical need for a serious or life-threatening illness. Notice the language, and there are some people who are very much engaged in the Pcast report now, and if I'm misreading this. I'm not. I'm a lawyer. I know how to read English. But if they didn't intend this when they wrote this, so be it. They can correct me. But the language does not say that accelerated approval was really good because it got important drugs to patients faster, though it did, or at lower expense, it did that too. What it actually says is we wouldn't have many of those drugs had it not been for accelerated approval. We allowed for the development of those drugs. I hope it intended to say that, because if it didn't, it should have. That is exactly, I think that is plainly right, and I want to tell you why. The key to accelerated approval, and we'll get into some detail shortly, and then I'll stop, is that it slashes big chunks out of the standard FDA trial protocols. And you can slash phase 3 or chunks of it. Sometimes they push lower, and some quite aggressive doctors are saying we should be slashing all the way back into phase 2 or phase 1. It's our molecular medicine is that good. But in brief, the accelerated approval works by loosening the FDA's quite throttling grip on the drug development process. It loosens it enough to give doctors the chance to use the tools of modern pharmacology to learn how to fit a drug to a patient's molecular profile. Drugs developed and with HIV, if you look back at that history, the actual targeted drugs, that got the nucleosides and the protease inhibitors and the different families of HIV drugs were developed very quickly. They were licensed super fast, and none of them, in fact, were going to do any good, not for long, not alone, because the virus mutates too fast, but it was doctors who began assembling them into cocktails, and which were eventually licensed too, but it was doctors who then took the initiative to work out how to use these drugs. Right, with cancers, as I understand it, and I can confess I have not done a systematic study, but all the literature backs this up. Basically, if you shut down off-label medicine in oncology, you would largely shut down oncology. I'm not saying it's everything, but it's a very large part of it. Basically, to a rapidly growing extent, the doctors take the drugs, use them as sort of molecular scalpels and sutures, and go in there working out what to do with them, sometimes patient by patient, and sometimes sequencing a tumor at multiple points, because there's more than one cancer, biochemically speaking, inside that tumor. And in the later stages of the disease, of course, it gets even worse. The example I like to cite, because it contains the most dramatic illustration of how strange things can get, is breast cancer. We know there are at least 10 biochemically distinct forms of the disease. Some are targeted with an estrogen blocker, and get this, some are targeted with estrogen itself. It's you need one for one class of the disease and another for others. The performance of one of the blockers depends on how the patient's liver metabolizes the drug, because it turns out a metabolite is doing most of the blocking, not the drug itself. So you get another dimension of patient selection there, and we have other classes of drugs for the HER2 receptors, Herceptin, and so on. Oncologists select by combining and mixing and matching off the shelf. Complexity in our diseases, and therefore in our drug science, I think is the new norm. There's a lot of complex molecular diversity lurking under the clinical symptoms of all cancers, almost certainly all autoimmune diseases and neurological disorders, and even common diseases like diabetes. They just aren't going to be cured by one-size-fits-all drugs, which is very unfortunate, because the FDA's standard protocols are pretty darn good for regulating, testing one-size-fits-all drugs. But I am persuaded that they are not just bad or slow or expensive, they are worse than useless when you are actually trying to cure complex, biochemically complex disorders. I think that conclusion is all but inescapable when you begin thinking about drug trials and what the FDA is supposed to be doing the way everybody else in the industry is already thinking, which is to say from the bottom up, thinking about molecules interacting with other molecules and trying to think through what happens in your typical three-phase FDA trial. Inside a patient's body, a drug is going to interact with a finite number of other molecules. In that molecular ecosystem, we hope, but we're not sure in advance, that the drug will do something that seems likely to have some good effect further along the pathway that connects the molecular to the clinical. For example, by shutting down one signaling channel that's propelling the tumor's growth. But other factors are very likely going to limit or undermine that good effect. Our bodies are resilient, and the diseases that hijack our body are resilient too. How the patient's liver metabolizes things, as we've seen, maybe another factor that is quite far removed from the actual target in the disease, but that can matter too. Or the disease's resilience, can be attributable to multiple signaling pathways. The 3 different sets of signals are propelling your cancer, and so blocking one, yeah, it'll help. You may see an effect, but not enough. Or the disease may be at the stage where its chemistry is changing so fast, the later stages of HIV infections or cancers, for example. Or the drug, and this is one of my favorite, works better or worse in patients who carry more or 2 copies of the same genetic variation, or alternatively, the drug is no better than a placebo because you actually have a whole class of patients who can beat the disease on their own. HIV has its elite controllers. And the problem with having patients who have their own placebo is it can really mess up your statistics if you're not aware of this. You can get quite the wrong results that way. And then, of course, once again, side effects compound this, add more molecules to the stew, which are going to determine how you fit the drug to the patient. So what kind of drug science do we end up with when we push the standard FDA testing protocols through to their bitter end, to test clinical safety and efficacy rather than to search for how to fit the drug to the patient? If your paramount concern is safety, it's perfectly straightforward. You want larger trials, you want patients selected more indiscriminately, and you want to run them for a long time. I mean, there's no question. That is the best way to catch every conceivable adverse effect or side effect of this drug. And you may well find so many side effects or some that are so bad that you stay safe by rejecting the drug entirely. If you're searching for efficacy, it's a more subtle search. You have to include enough patients so you give the chance a drug to meet the ecosystem that it likes, and can do some good for some patients. But the pursuit of high efficacy very quickly becomes a narrowing pursuit. You actually want to get selection criteria for your patient, narrowing down molecule by molecule. This is how I choose the patient to fit the drug. And it sounds harsh, but it's good medicine. You vote the wrong patients off the island. You get another drug for them, but it's not going to be this one. This is not, a conspiracy against unfortunate people. It's biochemical reality. So the bottom line is a trial that involves lots of patients, indiscriminately selected, will simultaneously find more and more side effects, and that after a tipping point will begin the drug will begin looking less and less effective because you're prescribing to more and more of the wrong patients. What do you do about it? You can't work out all the patient selection criteria before the trial starts. I mean, I think some people are saying, well, if the drug is targeted, we got the selection criteria, no more is needed. It's just not working that way. I mean, then you're back to the sort of magic bullet disease. Okay, we've got one marker. Sometimes you get that. Gleevec worked that out that way, as Alcori apparently did, but it's not going to be the dominant model of pharmacology going further. Even when you have a precisely targeted drug, you probably haven't explored enough of the variations in that EGF target, say, to work out fully how it's going to work. You haven't looked at the side effect markers. You just still have a ton to learn. You have to have a process in which fairly early on in the process you are looking systematically for the right patient selection criteria. Working out that objective molecular science is never sufficient. We are always going to have a big layer of empirical statistical correlation piled on top of that, but it is absolutely necessary. Your drug can't even get started or go a long way if you don't do things that way. Stated in sort of conventional terms, the clinically based patient selection criteria traditionally used in assembling patients for a trial is too crude. It is too far removed from the level at which drugs actually operate. And if you start with the assumption of complex rather than simple molecular to clinical links, you immediately begin framing an adaptive trial process that really looks very different from the ones that the FDA now ordinarily requires. Your starting premise is that a drug that we do need will be only one piece of a more complex solution and will provide only some and perhaps only temporary clinical benefit to some, often a minority of patients, not the majority, that have been rounded up initially for the trial. The conclusion that a drug is worth having will be often based on subclinical effects, sometimes as far down as the cellular or molecular level, a reduction in HIV or cancer cells in the patient's blood, for example, and the benefit may be a distinct minority benefit in 1 in 10 patients, and the other 9 just may need other drugs. Under the accelerated approval rule as currently applied, the FDA makes ad hoc calls. Its standard protocols, by contrast, allow nothing of the sort. And if you're going to do these, it's very clear that very quickly you actually have to get away from standard statistician scripted trials to to computers because you want to collect torrents of molecular data early on in the process. You may be looking into multi-drug regimens early on, and you are in search for a complex array of patient-side markers that can serve as the patient selection criteria. Rigorous multi-dimensional trials require modern statistical tools. We're talking Bayesian if it's your thing, and that's what the Pcast report says too. And you restructure trials on the fly as you learn, because that's how you develop Bayesian statistical confidence in your early hypotheses. The search should span both efficacy and safety biomarkers. We're developing catalogs of safety biomarkers very rapidly. We should be, when we see side effects, we should be testing systematically against those markers to see if we can narrow how a drug is prescribed. And the licensing of the drug won't be the end of the process, of course. All good Bayesian, I mean, the whole notion of rounding up a fixed number of patients and conducting a trial for a fixed time is an artifice of the current statistical methods. It's a frequentist artifice. That's what you do if you're a frequentist. Bayesians don't do that. You have an open-ended process. That doesn't mean you don't have to make licensing calls at various points, but it's not inherent in the statistics. The adaptive learning process, by and large, is anathema at the FDA. I know they've got a few of these under examination, but they aren't really. They've been waiting. They've had 20 years to do this. Enough's enough, and they should get on it. Now, the final thought is this, and I don't want to dwell on this, but the fact is, even individual doctors now are rapidly acquiring the tools, and at affordable cost. I mean, they're going to become very widely available, where they are extraordinarily good at tracking a disease's progress and a drug's progress or its effect inside the individual patient. And many of you know what I'm talking about. If you want to read a really most interesting demonstration of this, Stanford recently conducted a 2-year tracking of one of its professors and the senior leader of the team's biochemistry, 40,000 molecules, collecting billions of data points from the body of a team's senior member. I think it was on day 301 of the trial, they to their surprise, saw his blood sugar leap up. Okay, he had contracted diabetes. In short order, they tracked it back to a viral infection that he had experienced 12 days earlier, and then they zeroed in on the autoimmune chemistry that had actually kicked this off, and then they watched a few drugs and a change in lifestyle wrestle his blood sugar levels back down. And as he said, as the patient said, the Professor Snyder said, in a study like this, you are your own best control. You compare your altered or infected states with the values you see when you are healthy. I mean, instead of a placebo, you use yourself if you've been tracking long enough. It's ingenious. I mean, that kind of power, I think, is rapidly moving out. The biotech industry is giving it to us. And with that kind of power, you really fundamentally change how you begin thinking about the etiology of diseases and the prognosis, and also about what you think about the success or failure of drugs. A drug is one more molecule thrown into the stew, and if you can really track things in real time and get rapid feedback, you have an alternative regulatory tool in place. This has, I think, long-term implications for what the FDA's remaining role is, but for another day. Thank you.
Take some questions now, if you please. Raise your hand and identify yourself. Josh? And a microphone will come to you.
I'm Josh Bloom, American Council on Science and Health. Coming out of drug discovery, I just have a slightly different take on your point that the bottleneck is the FDA. I think it's the drugs coming out of Big Pharma that are the problems, and the problems that they've run into, multiple signaling pathways, off-target toxicity. Maybe it's my laboratory cynicism showing through, but I have a feeling that if the FDA did everything you wanted at the snap of a finger, it wouldn't make that much difference at this point, because the molecules that are being approved and they're going through the clinic really are not good enough to handle multiple variations and mutations in these genes.
I couldn't agree more. I think it's biochemically impossible to develop such a molecule. I mean, if the FDA, you arrive with a new colon cancer drug and the FDA said, oh, by the way, let's test it, but we're going to assemble 500 colon cancer patients and 500 cholera patients. Go for it. I mean, you'd say they were crazy, right? Or if you have a pneumonia drug and they said, well, we're going to give you bacterial pneumonia and fungal pneumonia, you'd say you're nuts, right? Why? Because you actually know they're different diseases. They're biochemically different. Well, the FDA is, where are we working out all of the patient-side variations that affect a drug's performance? The science, look, we know we can do better than the FDA. I mean, the computers, if the drug sneaks through somehow or other, the databases that arrive after do far better than anything you could possibly get out of an FDA label. It's the science isn't with the FDA here. We're not developing it.
So you actually think that with liberation of the process, you'd see a material difference in some of the survival rates? And
I am saying that drugs are molecules that interact with other molecules. We know how to get that science right if we're given a framework for getting it right. The key stage of doing that is in the FDA process. You can't do it all beforehand, and if you wait to do it after, there may be no after because the drug never gets through the FDA licensing process. So we need a process. This is not a statement, go away FDA. It really isn't. I don't think a single serious drug company in the country would back that because you lose, people need the confidence, and so we want an FDA. You've not heard me say abolish the FDA. You won't, all right? I think the FDA has the tools to do this. President Obama's administration says so. They're right. I don't say that all that often. This is Manhattan Institute, you know, but no, they've actually, two really excellent reports have emerged from this administration, and I am very heartened by this. I think there's a tremendous bipartisan opportunity to say, you guys understand the science. Your NIH, your National Research Council, or at least your people appointing the experts who are on that council, your Pcast, yeah. So, yeah. Oh, wait, somebody else calling you.
My name is James Shea. I'm a physician scientist at Memorial Sloan Kettering, taking care of.
You guys are doing fantastic work.
Yeah. So I totally agree with whatever you said, because this is not just the FDA problems. This involves insurance company problems. This involves drug company problems. For example, like we did research in a lab specific for one patient, and then I want to give this drug, and insurance company say, you need to go through these two drugs before you can even give this. And they will say, this is another drug. Nobody knows this drug should be approved for that. So there are many issues like that. The other thing, the other question really is, for example, we like to combine 2, 3 different drugs. It's just like, you are totally on the right point. It's like a virus, it's like bacteria. We need to use combination therapy. The problem at this moment is the drug company, they are not eager to work with each other because each company have their own individual good drugs, but we have trouble to convince them or through an appropriate channel to combine the drug together. And the other thing really get to the point of this is the biochemistry and all the omics that we know at this moment really explain part of what we know about the cancer biology. Yes. And that's why the pattern recognition instead of the linear code will be the way to solve the problem. That's what,
Look, I hardly, first of all, in case you don't know, this gentleman's hospital is doing absolutely pioneering work on this. I mean, you guys, unless I'm remembering the wrong source, are doing the multiple sequencing at different points in tumors and so on. I mean, why are you doing that? Because you know there's more than one disease down there and you want to hit them all. Otherwise, you probably haven't achieved that much. I think the general problem at the FDA is they can do simplicity. I mean, statistical correlations, frequent statistical correlations are very good when you have a single cause that delivers a single effect in a straight linear way. But when you have a whole network and a chain reaction, if you have a chain reaction, a flaw at any point in that reaction can give you the same clinical symptom. If you have a web, you can, you break up one chain and it bypasses and goes another. The main question is whether cancers and HIV are unique or whether lots of other diseases are like this. All of the evidence that's emerging from the human genome, the 1,000 Genomes Project and so on is indicating that we've solved the simple problems. We aren't designed as one-size-fits-all bodies. If we were, some microbe would have wiped us all out, a million years ago or something. So complexity does not get through this system. I mean, the FDA does not handle complexity at all well. And if we don't do complexity, we won't cure a lot of diseases.
Paul? Just wait for the microphone.
One thing I want to take exception with, you said the FDA does a good job of the simple case, but of course there's a whole huge literature by economists showing that even for that case, the FDA has been very, very cautious and has been very, very too slow and too expensive in approving drugs. So I guess it's not surprising that this newer technologies, they're not doing very well either, but I don't think they've done that good a job
all right,
in the past either.
All right. Well, I won't, far be it from me to argue. Look, in response to the early part of your comments as well, I only had 20 minutes here. If you want, look, obviously the money end is important. I think the intellectual property rights are extraordinarily important and insufficiently understood. And by the way, I think many doctors are instinctively on the wrong side of this. Make everything free, put everything on the network. The problem is people are spending $300 million to compile a data set, and you won't get that kind of private money putting that kind of money into developing these databases unless you have some incentive there. And the federal government, you may have heard, cannot afford to do it for all the genes everywhere. So I want to enlist the private money as well. The reimbursement strategies are a major component. I mean, I have some fear, I hope not vindicated, that basically cost control will become a reason for imposing another layer of all statistical, all empirical analysis at the top here, and will therefore find a second way to throttle the pursuit of molecular medicine.
Let's take one more question, please. I'm sorry. Mark.
Mark Levin from Third Rock. Peter, we talked about this a little bit last night. I agree with you. The National Research Council report and the Pcast report and your report are fantastic. I guess my question is now that we have these reports, what do we do with them and what's the process? Obviously, the FDA is changing the breakthrough product regulations, et cetera, but these things will take a long period of time. How do we coalesce all these reports and actually make a difference in an organization that actually may not have the right people and needs potentially a different political structure? So how do we take this report and make a difference?
Well, a few comments. First of all, I actually spent 25 years in Washington fighting a somewhat similar battle against the FCC. I arrived just as the transition from analog to digital technologies was arriving in the early 1980s. We had a 70-year-old agency that was regulating, telephony and broadcast and everything based on very technologies that were clearly going to be overtaken by digital. The writing was clearly on the wall. It was a very interesting process. We had to fight it at every level. We fought it a lot at the FCC. We took some cases to the Supreme Court and won them. We got major new legislation enacted in the 1996, I think it was, Telecom Act. We had to litigate a whole bunch of antitrust cases. You mentioned collaboration among drug companies. A good thing, necessary. The FDA has sponsored some of these. You do actually have an antitrust department as well, which does not like collaboration among competitors. So you have to explain to them that if there are 20 EGF blockers out there, you're not really competitors if you've got 2 different EGF blockers. Now, getting to the FDA, I am not an expert in political reform, but I'll tell you what my gut instinct says. First of all, I think there is tremendous potential in the explosion of cheap diagnostic technologies. I mean, we can get out to the masses. The FDA, by the way, is not at all happy about this happening unconditionally. Neither are insurers. There's quite a lot of pushback. But the fact is, we will soon be giving most every American who wants it the power to read all sorts of molecular detail about their code. And, if you look back at the HIV years, we made incredible changes in the regulatory process because without the help of the internet and without Twitter and all these things, the gay community mobilized and communicated and put enormous pressure on Washington. And some people say that was a disaster, a leap back to the future, not enough FDA. To the contrary, they actually pushed the FDA into the modern framework of doing a lot ad hoc. We licensed thalidomide thanks to that, because they started importing it from Brazil, and that forced people to work out the science and so on. So I think that's the first very powerful force. I think I'm very eager to see the digital community heavily involved because they're very rich, they're politically influential. They are loved actually on a rather bipartisan basis. I mean, the left likes them and the right does as well. And I think they play an essential role here and have something unique to offer. And so that Sergey Brin has kind of got a nose under the tent through his wife and 23andMe. I think it's good news. Come on, Sergey, do the rest, or help us. Yeah, there are others, but if you look at the political revolution across the globe, I mean, the microprocessor and the web did a lot. I think we have information is a very powerful tool here. Thank you.
Thank you very much. And we're going to take a brief, about 5 or 10 minute break before our next panel, so feel free to check your cell phones and get a beverage. Thank you.
I think it is clear how fortunate we are who believed in the power of ideas.
Supply the common sense and the fresh thinking of the Manhattan Institute.